Hello everyone. Welcome to this walkthrough to go over our web application to explore gene regulatory networks derived from single-cell chromatin accessibility and gene regulation data. Our goal was to create an interactive resource to query specific target genes, transcription factors, and the genomic loci where we predict the transcription factors are acting on these target genes. First, let me give some background on the biological system we're studying. The goal of our project was to study the cells of the murine hematopoietic system as they differentiate into specific progenitors. We sampled the KIT-positive selected cells from the mouse bone marrow. It is important to note, however, that this kind of gene regulatory analysis can be adapted to practically any biological system to study mechanisms of gene regulation and cell function. What we wanted to know was what key marker genes turn on and off in these differentiation pathways, and which transcription factors are making these cell fate decisions happen. However, it's important to note that these are very rare events, and they haven't sufficiently been captured to date. So to narrow down to the cells making these lineage decisions, we worked to develop gating schemes for single-cell omics capture. First, the HSC MPP gate uses previously identified markers that enrich for the hematopoietic stem cell and the multipotent progenitor cells. First, a lineage dump is then combined with CD117 positive and SCA positive selection, or the LSK fraction. And then cells are filtered to remove the CD48 positive, CD150 negative fraction, such that we're focusing on the cells that emerge from the HSC state. Next, to highlight the cells that transition from the multipotent status to the specified progenitor status, we developed a new multi-lin gate. This gating uses a lineage dump to remove specified lymphoid and erythroid cells. Then the known markers CD117 and CD34 are used to get to the immature cell states. And lastly, we remove CD150 positive, CD115 positive, and LY6C positive cells to remove those cells that have already committed to different myeloid lineages. The result is to narrow down to these cells that are in the process of making a lineage transition decision. Here is a high-level overview of the gene regulatory network analysis. To start with, we use the site-seq data gene expression and surface protein expression values to derive the 87 clusters using SC-triangulate to integrate reference labels, de novo ADT labels, and de novo transcriptome labels. From the T-seq, we take the gene expression and then map those cluster labels from the site-seq using a modified pi-harmony and KNN transfer method. Since we're only looking at the HSC MPP and multi-lin fractions, we recover only 57 of the most immature 87 clusters. We then take the chromatin accessibility information from the T-seq and then split the BAM file into 57 separate BAM files based on the T-seq clusters. We then call peaks on the individual BAM file sets and then merge those peaks to have consensus peak sets. To nominate the important regions for downstream analysis, we focus on those peak regions that are dynamically accessible in correlation with the gene expression. So we filter to the TAD boundary to nominate those peaks that are likely to interact with the transcription start sites, and then score based on the correlation of the pseudobulk peak accessibility and the pseudobulk site-seq gene expression value across the 57 clusters to filter to the peak set of significantly correlated peak regions. We then take the BAM file, first using the total multi-lin capture to build a bias model, and then build chromBPNet bias factorized models, which results in 32 clusters with sufficient read depth to generate these bias factorized chromBPNet models. Using the bias factorized chromBPNet models, we can then generate base resolution contribution scores, which predict the chromatin accessibility of the given regions. We can then aggregate each of the common Seqlets or short regions of base resolution contribution scores into contribution weight matrices. So these are clusters of Seqlet identified regions that have similar motif patterns. We can then annotate those CWM patterns using known motif annotations from the SysBP2 database, which allows us to link the transcription factor identity to the base resolution contribution sites. Then to get the Seqlet positions across all the peaks, we do CWM scanning and then scoring by taking the dot product of the contribution score value with the motif identity across the Seqlet. And that lets us build a library of all the Seqlet instances for each of the 32 pseudobulk clusters. We then score the contribution score values against the gene expression values with the correlation metric again to look for regions that are similar to the gene expression changes that are being observed. This lets us filter to those Seqlet regions that are highly correlating to target gene expression values. Thus, we have a TF gene expression that's correlating to a target gene expression value, which is linked by a region of chromatin that maps to the TF motif identity that is also correlating in contribution score value. We then can take and aggregate these score values to get a general TF activity score for each of the clusters. So this goes over all of our analysis steps that help us to identify the total gene regulatory networks, which is the sum of the target gene expression, the upstream TF expression, and the base resolution contribution score at that region. To help navigate the GRN application, we wrote this walkthrough PDF, which is available in the description. You can either start by selecting a transcription factor. Alternatively, you can select the target gene. And then once the target gene and the transcription factor are selected, you can select a Seqlet to browse Seqlet track. Alternatively, you can walk through a cluster selection where you look at the marker genes or TF activities or target gene activities and select those elements that way. You can also navigate to a genome browser session with the Seqlet contribution scores loaded and navigate at your own pace there. Each of the elements of the browser is downloadable as a CSV file, which we illustrate here with these download viewer tables. So to get started, we can click this link and that will take us to the app. So this is just a simple application to search and request different items from a server that holds the data and will return and help visualize the different elements of the gene regulatory network. To start with, we can enter a transcription factor in the first search box here. So for example, if I search for CEP, and you can see it'll populate with some autocomplete options that are available within the database. So we can select CEP epsilon and click Update Transcription Factor. And now the gene expression centroid values will be loaded for each of the 32 clusters that we were able to make gene regulatory networks for. And you can see below that under target gene correlations, we can see the target genes that the particular transcription factor, in this case CEP epsilon, is highly correlated to, ranked by that correlation coefficient value. Now to select the target gene, we can either select one of the items from this table here, and it will actually show us beforehand how many Seqlet positions in the genome we were able to find base resolution contribution scores for that TF that would predict it to interact with this target gene. So for example, if I go ahead and select RGCC here, for example, we can see that its expression is tightly correlated with CEP epsilon. And then after selecting both the transcription factor and target gene, we get a table of Seqlet positions Alternatively, what we can do is we can select the target gene using this second search box here. For example, if I go ahead and type in CLEC12A, this is a very interesting surface protein that we identified within our study. If we go ahead and update the target gene value, we can see now that the target gene updates with CLEC12A, we can see it's pretty highly correlated to CBP epsilon here, highlighted in green, and then it is now filled the Seqlet table with the Seqlets that are around CLEC12A that CBP epsilon is predicted to bind to. So if we go ahead and now select one of the Seqlet positions here, it will populate the browser with the Seqlet tracks for each cluster. So this gives the base resolution contribution prediction from the Chrome BPNet model for each of the 32 clusters at a specific site at which we found this Seqlet that matches to the CEBP epsilon motif within CysBP2, and the contribution score here indicating that these bases are particularly important for predicting the accessibility at this locus is actually correlating to the target gene expression, CLEC12A, which in turn is correlating to the upstream transcription factor expression, CBP epsilon. So this is an edge within the regulatory network that is being described here. So alternatively, we can also select the cluster, and we would do so by using this third search box here. So for example, we can start to type out one of the clusters. So if I just type B, it will become pre-populated the only cluster that starts with a B, which is BMCP. So if we update the cluster, then we'll load 3 tables at the top here to describe features of the cluster. The first is a marker gene table, so how specific a given gene is to this cluster in particular. MS4A2 is actually the most specifically expressed within the BMCP fraction. Then we also have a table to describe the TF activities. So this is the aggregation of those edges within the gene regulatory network within the BMCP fraction. So it will correspond to the expression of the transcription factor, the expression of the target gene, and the base resolution Seqlet dot product score that we calculate. And so each of those would get aggregated across all of the TF instances, and that would build this score here. So you can see these transcription factors are predicted to be most active within the BMCP cluster. Similarly, we can aggregate, as opposed to just by the TF, but by the target gene, and you can see which target genes are predicted to be most active. So it's interesting to see the MS4A2. So if we click MS4A2, and now we get populated with the target gene, so we get the TF correlations that correspond to MS4A2. And one interesting TF seems to be CBP alpha, which has quite a lot of predicted elements connected to it. So if we select that, then we update both the Seqlet table as well as the TF target gene correlation table. And now we can update the selected Seqlet here and see the Seqlet change as well. So these are all of the ways that we can change how we view each of the nodes in the regulatory network and analyze them. So lastly, you can see in the Seqlets that we provide the Seqlet region, the region that's displayed by the Seqlet, as well as the Seqlet to gene correlation, which cluster and pattern this particular Seqlet came from in terms of the CWM, and as well as the attack peak that this Seqlet fits under. So we provide download buttons for each element in the browser here. And if we go ahead and click this download button, download the table as a CSV file, which we can open in any given table editor and make changes to it. So for example, you see here, I had downloaded the Seqlet table. And This CSV file will be populated with each element of the table on the viewer. So that's everything. We hope that this viewer provides a valuable resource to query elements of the gene regulatory networks that we describe within the manuscript, as well as enabling access to the coordinates of these particular TF binding sites that could later be verified experimentally.